I'm Dr. Alison Sanders, and I'm a postdoctoral scientist at the Icahn School of Medicine at Mount Sinai, and I work in the Lautenberg Environmental Health Sciences Laboratory. My research studies preterm birth, or babies being born too soon, which affects approximately 12% of births in the United States. Babies being born too soon have increased risk of infant mortality and are associated with later life health effects, including poor neurodevelopment and respiratory effects. However, what causes preterm birth is largely unknown, and our laboratory studies molecules in our bodies, or biomarkers, which could indicate a woman's likelihood to deliver early. In my work at Mount Sinai, I study a class of biomarkers called microRNAs. MicroRNAs are active in our bodies on a daily basis and are responsible for profound changes in our cells. Today, I'd like to tell you about our work to understand microRNAs as potential biomarkers of preterm birth. In a study of pregnant women, we measured microRNA levels in the cervix during mid-pregnancy. The cervix must dilate during labor, and we hypothesized that changes in microRNAs in mid-pregnancy might predict early onset of labor and preterm birth. Typically in our cells, genes are expressed when DNA is transcribed into RNA. This RNA is then translated into proteins, and the proteins carry out cell functions. MicroRNAs can potentially interrupt this flow of cell signaling and disrupt or alter the proteins that are built. Through our study, we were able to identify a set of microRNA biomarkers in the cervix that predicted a woman's likelihood to deliver early. These microRNAs were involved in cell signaling processes that involve the body's immune and inflammatory response. If confirmed in other populations and studies, the microRNA markers we study could be used in early pregnancy to predict the risk of preterm birth. This could help physicians make decisions to prevent preterm delivery and perhaps lead to new treatments. Predicting when a baby will be born is difficult, but an important issue in children's health. Our study provides hope that we may be able to predict early delivery and create screening tools to prevent preterm birth in the near future.